Okay, so just before we get started, welcome to the third session. In this session, again, we'll have three speakers, 15 minutes to present, 15 minutes for questions and answers. We're streaming online, so again, I'll check with each speaker. If we finish a little bit early, we can wait a minute or two before you, or a few minutes before you start, as you wish, if you're waiting for people to join online. Our jury members in this session are Olivia, Nicola, and Kurt, so thank you very much to the jury members as well. And yeah, I think that's it. So our first speaker is going to be Pedro, who's going to talk to us about accelerometers on humpback whales. So over to you, Pedro, and good luck with your presentation.
Thank you. Can I start?
Yeah.
Hey, hello everyone. My name is Pedro, and today I'm going to present my project, Drivers of Accelerometer Detection in Humpback Whale Sound Units, Implications for Call Identification. The project that I developed with the supervision from Dr. Franz Jensen and Dr. Jeannie Shearer. And to start talking about my research, I have to say to you that cetaceans, marine mammals, they are very important for their ecosystem. They play crucial roles and provide a bunch of ecosystem services. So therefore, monitoring their population is essential when we talk about marine conservation. And talking specifically about cetaceans, these animals, they adapt themselves very well to life on the water. And one of these adaptations is the use of the acoustic channel for a lot of functions, such as feeding, mating, communication, and navigation. So different groups of species have developed different strategies. And when we talk about monitoring these species, it is also very important to take into account the sounds that they make. So one way to do that is through passive acoustic monitoring, also known as PAM, where we can record we can take sounds from a specific location and time. And with that, we can explore this data as much as we want. And one of these ways is regarding population estimation. And how do we do that? Usually, we use a technique that we call cue counting, where you basically take the detected number of acoustic cues produced by a given species, and you then take these numbers and you estimate the whole animal density in the area. But to do that, first we have to know the individual cue rates, which is basically how many sounds one animal would produce in a given time, and then to extrapolate that for the whole population. And one way to access that is through biologging devices, such as DTAGs. And with the DTAGs, we can attach them to the animal and record a bunch of data, the acoustic ones, and also like a bunch of environmental data, and then we can use this data as we want. However, when we talk about acoustic detection in these DTAGs, we have a challenge. We have to make sure that the calls produced on that DTAG is actually from the animal that we attach the device. And we have obstacles when we talk about the ocean, because we have the sound propagation on the water that works very differently, is way more efficient. We also have the behavior context of the species, because they are usually social species. So we can have animals around that will also produce acoustic data in the DTAG. And what scientists usually do to respond to this question is to use received acoustic levels on the DTAG. Also, they assess the angle of arrival of that sound on the tag, and also signal-to-noise ratio. But these techniques, they are still like have their caveats, like you have to account for the presence of conspecifics, as I say, also the thing with the sound propagation, but also near-field and body shading effects that would be mainly related to the tag placement. But going now specifically for misty sets, one approach that is being used for caller ID is a type of sensor named accelerometer, which is basically a sensor that is inside the DTAG. And this sensor is actually measure the movements and like that the animal is doing. So it's mainly used in this kind of like behavior studies where you want to access that. But they noticed at some point that this device was also capturing the vibrations that the sound that the whale was doing. It could be detected also in the accelerometer. So here we have, for example, the acoustic spectrogram, where you can see the call, and here you have the spectrograms for the different axes of the accelerometer, and you can see some level of detection. So this was replicated for other low-frequency species. However, for mid-frequency species, such as the humpback whale, we just had one study kind of like describing that this detection is possible, but they didn't go further on that. They did investigate how this detection the characteristics of it. And they just mentioned that like some calls, some of the units, they were being detected by the accelerometer, but some they were not. So it's still unclear, and that's when we come. Is where we come. And the objective of this project was basically to investigate the accelerometer detectability in some units of humpback whales. And here are three main questions, but basically we are looking at the three different parameters, which is the unit type, how the unit type would influence that detectability, also the frequency of the call, and also the intensity. So why choosing humpback whales? First, it's important to know that just the males are producing the songs. So females and calves, they don't sing. It's just the males, and this singing is mainly related to the attraction of females, but also to interactions with other bulls. And when we see the structure of the songs, they have a very intricate structure, because they have subunits that turns into units, that turns into phrase, and then themes, and then songs, and then when the whale is repeating these songs over time, we have the whole song session. So it's a very valuable data to investigate how acoustic parameters are related to the accelerometer detection, because they vary a lot. The data that we used in this study was collected in Exmouth Bay in Western Australia, and they approached the animal with a pole, and then they just tagged the animal. And in our case, it was a sport male with next to a monocalf pair. So we give strength to the assumption that the song units presented on the detect were from this male, because the other two individuals, they don't sing. And we use the detect for with high sampling rate accelerometers in this study. And in total, we had almost 14 hours of recording. And the first thing I did was go through all of this recording and label all the song units that I was finding. And here we have like a visual representation of that. This is the whole recording. And you can see that the male was singing almost for 7 hours, like ininterruptedly, like all the time. And here's like just one of the dives that he did. And you can see like calls everywhere. And this is just another part, like a zoom of a small part, where you can see the different song units here. So the first thing we did after labeling all of them, all of the song units, we went through them one by one, and we were assessing the acoustic spectrogram, and then the spectrogram for each axis of the accelerometer. I'll explain later what are the axes. And here we divided them in 4 categories, this detectability. Here the detectability, where you can see clearly like this pattern repeated on the accelerometer spectrograms. A faint one, where you can see a lot of background noise, but you can see still like a little bit of the structure of the call. And absent, where you don't see at all. So in total, we had 64% of all units presenting some kind of detection, either clear or faint. Later, we went to a visual inspection of the spectrograms to classify them according to their signal type, to answer our first question. Then we divided them in 3 categories for that. We have tonal sounds, pulsive one, and mixed, where you have kind of both structures on it. And now you're gonna hear each of these sounds like one after the other, and you see how they sound, and you can see they are very different. So as you can see, it varies a lot within the song units. And to do this visual inspection, like it would take a long time to go through one by one. We use BirdNet, which is a software that generates deep learning-based embeddings. And then these embeddings, we can group them based on their similarity and make kind of clusters. And we can project them using TESNE, which is also another tool. And we have something like that. As you can see, we have a big variety of song units, and I can say this guy deserves a Grammy. And in total, we had 18 different clusters, and we have significant results when we talk about detectability in the accelerometer and the clusters. So we did see a difference in the detectability according to the song unit. And here is going further on that. Here we have this graph representing the three types of song units, so tonal, inclusive, and mix. And here each of the clusters that were inside each of the song units. And we also had significant results for the sound type. So here you can see that tonal units, they have way more detectability compared to the PUC and mixed ones. So we can say that indeed cloud structure is a key driver of accelerometer signal detectability. And we believe this is because of how the energy is spread when we have in the units. Like when you have the tonal ones, the energy is more concentrated, while when you have the pulsive and the mixed ones, they are more broad and like they are very broadband. So the energy is kind of more dispersed, and while here is more concentrated, and we think that could be the reason why we have more detectability in the tonal ones. But here one interesting thing is that even inside the same cluster, we have big variability of like detection. Like in cluster 4, for example, you have 20% of clear signals, 60% more or less of faint ones, and then the rest, no detectability. And this makes us think like, what is happening? And then after this, we went through fine scale analysis where we went to the two other questions that we proposed. But to do that, now I had to go through all of the units, but then we filtered just by faint and clear, the ones that we had some detectability. And what I was doing is what we call time frequency bounding. So we basically put this square in the unit, and we use a code in MATLAB to extract different parameters, acoustic parameters. And we also have this noise segment to calculate signal to noise ratio to see if like the background noise is interfering somehow the acoustics. And here we have the first results, the second results for the second question, which is relating the call intensity and the accelerometer detection. We perform a GLM and we see significant results for that. So here you can see that clear signals, they are more concentrated when we have better intensity call intensity. So we can say that call amplitude is also a key driver of body-borne vibration in this way, in this case, and that the accelerometer signal is scaled with the sound pressure level. Then we go to frequency. We also, to go to frequency, use the peak frequency, which is where in the frequency we have the most powerful levels, the levels of power. And we divided these peak frequencies in 4 categories based on ranges. So here we have the lower ones going to the maximum of 500, which is the limit of the accelerometer. And then we perform also GLM using these categories. And our focus here was in the main effects, and we could see that the calls with lower peak frequencies, they were producing higher accelerometer levels. And this would go down when you go up on the frequency. Here where you can see in the regression line, this is the first one and the second, and this one is the lower ones, this one is the highest. And so yeah, low frequency calls would be more reliable to produce stronger accelerometer signals, and we believe this could be explained by how the whale recruits different types, different structures of the larynx to produce the sounds. And the last results that we also analyzed here, we took this chance to analyze, it was about the differences in detection in the axis. So I told you about that I would explain about the axis. Basically, the axis, they go for, they measure the direction where the energy is being spread. So you can do like that. First we have the z-axis that is like vertical, then we have the x-axis and the y. And in our case, we noticed that the detection were being more present in the x and the y axis. We did a pairwise t-test, and actually we didn't see a significant difference between these two axes, but with the z, we saw that. So basically in Norway, like the vibrations during the singing, they would be strongest in the horizontal planes. But this is just one way when they were like, okay, maybe could be explained by the tag placement. So it's still something that we have to give more research on it. We have to research more. So as a conclusion, we can say that accelerometers in DTECs, they may not be fully reliable alone because we had just 64% of detection, but they could support Caller ID as a complementary tool, as they were. We had, for example, some units with good detection, and these results that they are very interesting because they were the first ones in this type in this species, like with some more description. And for future ones, would be interesting to tag more animals and like also process their data. Also maybe try to check for variety tag placement. So maybe like tagging the animal here with the one tag and the other tag here or here, and then check how it differentiates the difference between them. Also adding another vocal context such as foraging calls and another type of sounds. And of course also explore other species. And that's it. Thank you very much. That are my references here, and I want to thank you, like all the team that did the tagging and all the funding and everyone that was present in these 2 years of master. My friends in Denmark, my friends from Garcia, my friends from Brazil, and that's it. Sweaty.
So I pass the directors for
Well, I can start. Thanks for the presentation. I can't really understand how this will help. You said that DTECs would take signals from other animals. What was the new thing here that you could avoid that? I didn't really understand.
Yeah, basically, what we're testing here in this specific study is how the detectability is related to like acoustic parameters.
Yes.
But like, for example, in this project is part of a bigger one where they're testing, like for example, if you have two whales here together and they are both producing the sounds, the accelerometer from the DTEC of one of the whales would capture this production, while the other one wouldn't, you know, like from the other whale. So in this case, what I bring is mainly like the relation with the acoustic parameters. But like, for example, I have my co-supervisors and my supervisor publishing an article where they actually made this interaction with like two whales and seeing like if the DTEC in this one would capture also in the accelerometers the calls from the other one.
So you're pretty sure that the signal that you have
is from the whale, yeah.
And what about here you have one animal?
Yes, one animal.
So how much can you say from the study of one animal?
Yeah, that's something that like also was like I was thinking a lot during my research and talking with my supervisor. And actually, it's just one animal, but like we had almost 9,000 calls with all call units, and that's a very high value compared to other studies that you have, for example, 3,000 ones, and like we have a good number of calls. And unfortunately, with the time, I just have 5 months to go through other animals. It would be very difficult. In the beginning, I even started like doing the part of the labeling. I labeled other animals as well, but then when we went to do them, like some of them were not producing sounds. They were basically producing other types of songs that we could not affirm. Like in this case, we have the assumption that this male is doing the acoustic register. But yeah, of course, like one animal is not the idea. We want to put more data on it for sure.
If you speculate, do you think you would have the same kind of result?
Yeah, probably yes, because I mean, if you use the same type of text and the same context, which is singing, I would say probably yes, because usually the males, they are like stationary singing, so they like are in like kind of the same position and like they don't move a lot. So it depends also like if there are individuals around or not. So Yeah, I think we, of course, if we bring more data to it, it will be more stronger, for sure. And it's actually something that we're thinking about doing like for publishing. But unfortunately, like with 5 months, like this took so much longer, like to go through all of these and do all these analysis. So yeah, fortunately, the future, yeah.
You do have from other stuff in your data.
Yeah. And some of them were not actually like the lower we had lower sample rates from some of them, so we couldn't go through the whole like frequency range. So we focused on this one that we had good results. It was singing a lot, as I show you, and we had different units. So yeah, but it would be nice to bring more individuals, of course.
Yeah, thank you for the presentation. I guess a few questions. So you mentioned the TSC, you get a representation of the different type of calls or patterns, within, so basically for the one individual type. And I was wondering, all those different goals, why did you decided to cut the signal and say, okay, that's one unit, this is another unit? What did you do that?
Basically basing on articles, like mainly this one, because there they describe the whole like structure of how the songs of the whales, and then there they explain about like how to differentiate the units and this kind of stuff. And so I basically based on literature to classify this.
So you did it like by hand? Like,
Yeah. Yeah, you open the whole recording on MATLAB, and then you go through all of it, and then you pinpoint the beginning of the call, the end of the call, and then you kind of label them, and then you extract, and then you play with the data.
Okay. Yeah, I guess I'm gonna go more general questions. I didn't really understood so it took me a while, maybe because I'm not familiar with the subject, but to understand that. So the accelerometer you get, when I saw the title, I was like, oh, okay, this is for movement, where it's actually for the sound.
Yeah.
And I was wondering what's the difference between the information you get between the XYZ axis, where you decompose the direction of the movement of the sound, of the sound waves, and what is the signal? Is the XYZ axis just a simple decomposition of the higher acoustic signal, or there is more, or is it some sort of difference?
I'm not sure if I fully understood, but can you repeat like that?
On the first graph,
yeah.
the signal is just on the first graph of the.
Is the acoustic?
Okay.
Yeah.
How does that relate to XYZ? Is basically the acoustic signal you get on the first axis is basically made out of the three following ones?
Yeah, it's coming from the acoustic one, and then it's reflected on the three axes. And it depends on like how the energy is being like transmitted, the direction transmitted. So like you have the, like I told you, like I show you, you have the different axes, and then, yeah. So actually, it's very interesting because we're expecting actually the z-axis, for example, to be stronger. Because if you have the structure inside the way you're producing the sound, we were expecting the energy to go vertically outside. But we noticed that like the X's and the Y was going like, we were being stronger. So this kind of, these bodyboard vibrations that were spreading more than we were expecting. But like, we come to that, this is just the data from one way, and it would be nice to have other types of tag placements to see like if this will still be happening, you know?
Yeah, okay. And so I was wondering how is this information relevant or important, given that if you fail well in the wider context of your study, you can be able to have attack on maybe one web, but decipher only webs that are around. So the specific orientation of the code you get from the web is going to depend on the placement of the attack. But in terms of XYZ axis, like the code for a completely different web will not necessarily be related to the position of the sensor. So I'm like, what kind of information do you have in the XYZ components that is ecologically relevant and that you don't have in the Go web?
Yes, basically what I can say is that the main thing here was like to focus on like individual coup rates, and like for you to measure them, we have to know that the animal is producing, right? The call, yeah. So when I presented the different axes, it's just to show that like this type of detection also varies between these axes, and I'm trying to relate this to your question, and I'm not sure if I understood correctly. Sorry.
It's just, generally, but the question is, how does those XYZ axis allow you to tell, in addition to the simple signal, about the ecology of the
Okay. No, the XY and axis is mainly like, for example, I told you that the accelerometers they are used on the other types of studies like related to behavior. So, for example, if the whale is doing a behavior that like do like that, we can access that through the different axes, because like if she does like that, then this axis is like taking more and more. It would be producing more stuff. If she does that, she like the animal does that, and or like do that. We have different measures on the axis, so we can relate that to specific behaviors. And yeah, that's mainly like where these axes are being used in literature. But then here we just wanted to explore like if this energy is differentiated between them, you know, and like how they are.
It's interesting to think different approaches, but it's interesting that you're using something that should normally be noise if you're using accelerometer to do something else. I'm wondering, so you can confirm some of your ideas because you get some overlap between the acoustic and the accelerometer. Do you think what you see, and I don't know much about this, but do you think those calls that you can confirm are from this specific individual, and not just in the context of one individual, but in the context of multiple? You think that these confirmed calls, you know it's that individual, could that help you for all these other calls for which you don't get the accelerometer recording? Like that you only get the acoustic signal? You think that you get what the kind of data or the pattern or whatever could help you confirm or better ID under these other calls for which is 30% or something for which you don't have the accelerometer? Yeah,
like this is, I think yes, and especially like as we see here on the first results of here, we have specific type of units that have really good detection. And then like, as I told you in the end in the conclusion, the accelerometers alone, they might not be reliable because like 65%, 64% is also like, is not like the desirable number to use that to extrapolate like for all kinds of acoustic sounds, for example. But for some units specifically, yes, we could do that based on, I think so, based in the results we had. Yeah.
Thanks, Pedro. Lots of comments.
Maria. MLNT. The fourth. Maria.
This one?
Old, no, the new one. I guess.
This one.
I guess, yes. Let me check 1 second if it's the correct one.
if you just go into presentation mode, yeah. Okay, so moving to odontocetes, I'd like to welcome Maria Del Mar to talk to us about your thesis presentation. I'll wave at 2 minutes, I forgot to say that at the beginning,
Sure.
and then I'll stand up if it reaches the time. So over to you, Maria. Good luck. Yeah.
Thank you very much. Well, my name is Marlene Salmeron, and I'm going to present my thesis about identifying signature whistles in male bottlenose dolphins, also supervised by Dr. Franz Jensen in Aarhus University. Well, we are living in a biodiversity crisis right now, and this biodiversity is declining at unprecedented rates. For this, we actually need tools that are effective, scalable, and non-invasive methods to monitor populations. One of these tools that is a new thing in bioacoustics is the acoustic individual identification, or also called AID, that basically identifies vocal features of individuals, unique vocal features, that without the need of physical tagging or direct observation, which are the traditional methods for identifying cetaceans. This is a tool that is very interesting since it can allow us to monitor over time these individuals and over a range of space. And it's important, and also it's good that we use bottlenose dolphins since they produce vocal individual vocalizations that are known as signature whistles. These signature whistles I basically use for, it's like our names, so it's to identify other conspecifics or even recognize them. Yeah. But identifying these signature whistles, it's not that easy in a vocal range of a recording, and in this case, it's very necessary to identify these individuals in mark recapture techniques to be able to do this monitoring over time. And in 2013, Yannick et al. Proposed the SIG-ID method, which basically identifies signature whistles through repetition patterns, temporal patterns of these type of whistles. To meet this criteria of the SIG-ID method, basically we need for minimum of 4 whistles in each category, and then 75% of the inter-whistle intervals to be between 1 and 10 seconds. But this method was validated with few individuals and only with moms and calves, and therefore that's why we decided to use male bottlenose dolphin to have a demographic range, better demographic range. Well, the main aim of this study is evaluate different whistle classification methods and detection settings to identify signature whistles in male bottlenose dolphins. How did we do this? Basically, we compared 3 methodologies or approaches. The first one was using visual classification of signature whistles, and then we wanted to compare it to an automated way of classifying whistles using the logarithmic artwork. And then we also analyzed to bout level using the visual classification to see if this type of analysis could be more efficient and faster than processing all the recording. But let's explain what a category and what a bout is. A category basically is a group of whistles that have the same contour frequency shape of the whistle. So basically we group all of the whistles that are in the same recording and have the same shape into one group. But then we have also bouts that basically we look at the temporal pattern. If we have a group of the same type of whistle that is that has the interwhistle interval between 1 and 15 seconds, we can group them into smaller groups and therefore without the need of analyzing the entire recording. So let's jump, when we have all of this clear, let's jump to data collection. The collection was made in Sarasota Bay, Florida, where they have a resident population of bottlenose dolphins that has been studied since the early 70s, so we have a lot of data on that. To these populations, they do health assessments annually, which is impressive. And basically, they do capture-release sessions with the same net, where expert veterinarians do these health assessments, and also they put hydrophones with suction cups on the melon of the dolphins so they can record the signature whistle. And therefore, we have a signature whistle database of that population of dolphins, which was great for our validation of the method. And then, after having all these signature whistles, we also attach a DTAC, like Pedro explained, that has 2 hydrophones and accelerometer data as well. After attaching the DTAC, they release the dolphins, and these DTACs can be attached till 24 hours, but because of the movement of the dolphins and rubbing between them, normally it drops earlier, and it floats on the water. So then researchers can track it using VHF radio tracking. And let's go to the data processing. Well, first of all, when we receive the recording, we go through it, and we need to see if the whistles or like the vocalizations were made by the own dolphin that we are following, or they're conspecific or the alliance made. In this case, we could know that was from the focal individual calculating the angle of arrival of the whistle. Since we have 2 hydrophones in the DTAC, with the time difference, we can calculate the angle, and therefore we can know if the whistle is produced by the focal individual. And then we had to label all the whistles, and we did that using the Sarasota Dolphin catalog that we already previously had of each individual. So therefore we knew the signature whistle of each individual, and we only had to compare whistles to the catalog to be able to identify signature whistles or other types of whistles named non-signature whistles. After that, we extracted all the whistles into WAV files, or audio files, basically. And we started with the classifications. So first, I'm gonna talk a bit about the automated classification with artwork. Before running all the whistles through this algorithm, we had to manually contour each whistle. So the algorithm could compare whistles and manage to group them with similarity of the frequency and the contour. Then we did the visual classification, where an expert analyst basically classified the whistles in each category. Some were signature whistles, but the non-signature whistles had different categories as well. Then once this analyst did the classification, other experts would also do it again, so we could compare different experts' results. And then from the visual classification, we extracted bouts of whistles of each category to be able to do the bout level analysis. Here's a bit of the data overview that we did. From 10 DTAC deployments of 6 male dolphins, we basically analyzed 1,021 focal whistles in 45.6 hours of acoustic data. For the visual classifications, we obtained 468 signature results in 10 categories and 733 non-signature results in 37 categories. And then for the automated classification, we had 526 signature results in 155 categories. This is important, these numbers. And then 675 non-signature results in 367 categories, compared to the visual one. And then for the bound level, we extracted 84 signature whistle bouts and 78 non-signature whistle bouts. After we did all of this, we proceed to see the performance evaluation of each method. How did we do this? With precision recall curves to evaluate how the different parameter combinations could affect the performance. But what is precision and what is recall? That's a nice question. Precision basically sees how many signature whistles were actually true signature whistles, and then recall looks at how many of these signature whistles were correctly identified. Here is a demonstration of different PR curves. In this case, this is for the visual classification. What can we see here? We have precision in the y-axis and recall in the x-axis. And then in this case, each curve would be a different minimum number of whistles that a category needs to have. And then each number of each point would be the upper limit of the inter-whistle interval range of the upper limit, sorry. And then the percentage threshold for each of the inter-whistle interval ranges, it's fixed always at 75%. We calculated different types of percentages, but these are the curves that were doing better results, so that's why I'm showing these ones only. The best parameter combinations, as you can see, circled in red, were when we had a minimum number of whistles of 8, an inter-whistle interval range between 1 and 10 to 11 seconds, and the percentage of 75% of inter-whistle interval range. Then, for the automated classification, we had different results, of course, and it's very important to see that the recall is actually very, very low, extremely low. However, the best parameter combinations that we had in this setting was a minimum number of whistles of 6, and it's up there, with an inter-whistle interval range between 1 and 6 to 8 seconds, since are all clustered there, and the percentage of 75% inter-whistle interval in this range. And then for the bound level analysis, we had better curves, also with recall higher. And in this case, each curve represents the upper limit of the inter-whistle interval range, and each number in the dots of the curve is the minimum number of whistles. We chose the minimum number of whistles to be swept around because we thought, since it's bound level and it's grouping in smaller groups, that this parameter might vary more the results. So that's why we use this. And in this case, the best parameters was a minimum number of whistles of 5, the inter-whistle interval range between 1 and 10 seconds, and of course the percentage at 75% of inter-whistle interval range threshold. Now that we've seen all the methods, I wanted to compare them to see how good they perform and all the things. And visual classification takes a long time to be done and classified. Basically, it's not on a scalable to large data sets too. However, it gives actually robust category definitions And in the performance, it has high precision and moderate recall, which is good because the signature whistles that are identified are for sure signature whistles, but it doesn't identify all the signature whistle categories. Moving on to the automated classification, this algorithm was very sensitive to contour variation. So if we had a signature whistle that had a little variation, it would put it in different categories. That's why we had so many categories in the automated classification, which this translates into low recall and variable precision depending on the parameters. However, despite all of this could be a potential tool for automating and scaling the process of identifying signature whistles. And then finally, going to the bow level analysis, this performance depends actually on whistle density and temporal repetitions. And therefore, in populations of dolphins that actually don't whistle that much or they don't whistle that often, this tool might not be the best approach. However, we had a moderate recall and high precision as the visual classification, and it's actually an efficient and scalable method to identify signature whistles. And let's go a bit deeper in the bow level analysis. As I just said, our results just suggest that bow level analysis could be a promising approach for efficient scalable signature whistle detection in wild bottlenose dolphin recordings. And actually, it's being used by some researchers worldwide instead of the original SIG-ID method, but with the parameter values of the SIG-ID method to be faster and not use that many resources into data processing. In this case, Longer et al. Used the same parameter values, and Papal et al. Changed a bit one of the parameters, the minimum number of whistles, to 6 to be more conservative. In conclusion, we compare visual, automated, and bow level classification method using known source data. The bow level analysis matches visual and classification in precision while reducing the manual effort of processing. And this could be a groundwork for future development of scalable real-time monitoring technologies. And finally, this is a study basically offers both a methodological framework and performance benchmark for future long-term, non-invasive monitoring of bottlenose dolphin populations. I want to say thank you to all the team and everyone that's been supporting me, and especially to my supervisor for all the guidance and all the support. And, well, any questions?
Good. Okay. Thank you for your very nice presentation. What do they know about the dolphins? Do they have a signature? Well, do they have groups of animals that have the same signature if you go to another area? Because if they learn, maybe how to what do you know about that?
So yeah, signature whistles are learned since they are born. It works like kind of our name. So basically, each individual has their own signature whistles, but it's true that, for example, in case of males, they in this area, they go in male alliances, so it's two males together, and they have similar signature whistles, but it's not exactly the same. That's why it's an interesting feature for identifying them. And as far as I know, in each population, they have their own signature whistle. So each individual have a different one.
But can you find the same signature if you go to one area and then another? Because they have the same name.
it could happen, I guess. I don't think it's been described, but basically for that we need to compare both areas, and I think no one has been comparing signature wizard from one area and another because this is a very new kind of work.
Thinking about it,
Yes.
what you use your method that you define the videos and then you have two populations that are each other.
I could see that, for example, let's put an example in the Mediterranean Sea, that's where I'm from. If I had hydrophones in the Balearic Islands, where I'm from, and in France and in Italy, and the dolphins are moving around, then we will have the same signature whistle because it's the same dolphin. So that's one of the interesting things about studying these signature whistles, that it could allow us to monitor them throughout a range of time without just seeing them.
Thanks for the presentation. I have a question on your automatic classification, the art form. As you said, the number of categories it outputs is ridiculous because it defies the purpose of classification, which
Yes,
is as many categories as observations.
Yeah,
And is it completely unsupervised, or can you fine-tune the?
I believe it's unsupervised. However, we put the percentage of similarity that we want. In my case, and following the things that they told me in the person that created this tool, we use 96% of similarity.
To be in the same group.
To be in the same group. That's a lot. Also, especially because signature whistles, for example, have variants that basically contouring them and only looking at contours are undetected and therefore classified separately, even though we know and visually we classify them as the same thing.
And did you try to play around?
The thing is, we didn't have time.
How long does it take to run something like that?
Well, it depends a lot on the computer power that you have, first of all. In my case, for example, a group of whistles with 200 whistles, it took 2 hours, 3 hours.
Okay. Yeah.
But if I had one with 56, it would take minutes.
Yeah.
Because also we basically do iterations to.
Yeah.
I will try to explain it, the entire process. So basically, we put the contours, and then the artwork puts the whistles in neurons, and then in my case, I put 100 iterations, which we never reached that. Normally, it would stop at 5 or 6. Yes, and it took 2 hours. So imagine 100 iterations. But basically, if in the second round sees that it could reallocate some whistle, it would do that. Till no whistles was reallocated in another neuron, and then we had the categories results.
And I'm wondering if you lower the threshold,
Yes.
it might run faster, because it might converge faster.
Yeah, well, I, just out of curiosity, I did for one folder, just to see how it changed. I went to 90 and 94%. And of course, it didn't classify that much, but either way, it was actually a high high number of categories.
Okay.
So, yeah.
And maybe last one, maybe I missed something, but I don't. For the bound level analysis, you need to have visual classification first, right?
Yes.
How is it then scalable if you need to do all the visual annotation and everything?
Well, okay, we needed we needed the visual classification to extract the bounds. However, in real life passive acoustic monitoring, we can go through the recording and see that it's about, because we can see the seconds.
Okay.
So we can see that it's about, so basically extract that bout directly and then do all the parameters, check that all the parameters check to be able to see if it's a signature whistle or non-signature whistle.
To identify just the path, it's not, you don't do the initial classification like in the first example and then apply and then do the, you identify paths.
Yes.
Right?
In our case, we did it this way because we already knew that this category was a signature whistle and the other were non-signature
Now
whistles.
I understand.
We did it this way, but in real life, we don't have a catalog to see, oh, this is a signature whistle. So, yeah.
Thank you very much.
Thank you.
Okay, I guess you still my question. Yeah. I was thinking, I guess it's a broader, more general question.
Sure.
But so you are mentioning that you need some non-invasive technique and doing some passive acoustic is good for that.
Yeah.
And I was wondering, so I don't think we have signals at your products, so I think it's okay. But I was wondering if you, there is quite an intensive, in a way, capture process to tag documents before releasing them.
Yes.
And I was wondering which, was it to be disturbing, or was it defensive to be because of perfect passive monitoring?
Yeah, so in this case, we had already the database because they are doing health assessments every year, and that's why we are using, and of course this is a non-invasive, it's invasive actually, sorry, to the dolphins. I was not there, so I cannot tell you if they were disturbed as much or not, but from what I know or I've heard, they are already used to it.
I guess so.
Yes. So it's not disturbing. And they just go release, and sometimes we have some dolphins that actually start whistling immediately as they touch water, and some of them they are a bit more shy and need a bit more of time, but they do normal life. But of course, as I said, in real life palm detections, we are not attaching tags to dolphins. That would be a bit crazy to do. So we just have hydrophones everywhere, and we could just identify signature whistles.
Okay, thank you very much.
Thank you very much.
Okay, so now we move to fish, and I pass over, whoops, and pass over to Gregor for your thesis presentation. Good luck.
Thank you. All right, so we were speaking about mammals. Right now we're going to move to fish. I'm happy to present right now my topic, that is combined effect of anthropogenic noise and warming on courtship behavior in the painted goby. This guy here. I was working with Clara Morin, Manuel Vieira, and Ana Faria, that are my supervisors. Thank you to them. All right. First of all, quick overview of what we're going to go through here. Introduction. So I want to talk to you about the context and the knowledge gap of the study. Then I'll move on to the material methods, where I'm going to talk about the species, yes, the experimental design, some of the variables we are going to look at, and then eventually some statistics. Eventually after this, we're going to go through the results, and we're going to finish with the discussion and conclusion of this thesis. So, introduction. This thesis was done in the context of anthropogenic pressure on the ecosystems. So, as you may know, probably humans are putting a lot of pressure through different means on the ecosystem, on the marine organisms. We have habitat destruction, noise, warming, I mentioned already, pollution. All these are threatening species. We know about mammals, or that are suffering from noise. We know about sea grass and kelps that are suffering from worming, and fish and all that, of course, are suffering as well. Specifically, worming has been known to impact fish differently. So you know that warmer temperature means increased metabolic rate, and then increased oxygen consumption, which of course leave less energy for a lot of behaviors, or for example, for whatever moving or reproductions. The point here is as well that warming is influencing reproduction, and that's one of the main points of this topic, courtship and reproduction. Noise, this time, is also known to affect fish, and it interferes with auditory perception. We know about auditory masking, so the noises are interfering with the potential signal that they can get from other fish conspecific. As well, we know that noise is triggering physiological stress, and we know that, for example, cortisol is higher in fish that are put under noise treatments. And finally, it's also known to impair key behaviors. I mentioned reproduction, but it can be as well communication, it can be prey detection, and so on and so forth. So I talked about warming, I talked about noise. Separately, they have an effect, it's already proven. Here, we are interested in how these two stressors can combine, because actually we know that systems are put under different stressors, not separately, but simultaneously. And we're interested in how they can interact. Maybe it's an additive effect, maybe it's synergistic, maybe they cancel each other, so it can be either of them. So that's the gap here that we found no literature on that, even though noise has been correlated with other stressors. So here, the objective related to my study was to test first the individual effect of these two stressors, even though it has been shown. It's always good to try to evaluate this in this study. As well, we tried to evaluate the interaction of those, of course. And on top of that, we tried to explore relation between courtship, behavior, sound production, and eventually reproductive success, spawning. So yeah, that's it for the introduction. Now I'm going to go through material methods. I'm going to talk to you about the species, the design, behavior, and acoustic variables, and then statistics. So here is the species, the guy I've been working with for 5 months, very cute one. It's right here. It's a painted goby, Pomatuscitus pictus. It's a very small fish, it's up to 6 centimeters, and it's a benthic one. It lives on the sand substrate. It's polygamous. What makes it interesting is that males, they will build nests, as you can see on the left part here. This one is a natural one, and here you can see an artificial one that we are using in our study. That's very interesting, that will come back later. As well, they make sound, that will come back as well later on. They make thumps and drums. On top of that, they make a lot of behaviors associated with courtship. We'll come back to it later as well. Once the female decides to mate, the male will take care of the eggs only in the nest. And maybe you ask yourself why we choose this small fish for the study. Actually, it has been studied before in many studies, so it makes it a good model. It's easily kept in the control experiment in laboratory conditions, and it uses many behaviors in a breeding context. All right, that's the design. That's how we managed to test that. So we have 6 tanks, 6 aquarium. We put in each compartment. So there is the first compartment where the noise device is used. It's opaque, so there is no visual interaction with the fish. Female is in the center, male is in the right. You can see that we put the fake nest. I'll come back to it later, it's very important. Noise treatment, we use control, so it's nothing, no device producing anything. We have the speaker that is reproducing noise from both, in red. And then we have an intermediate treatment that is a coil. So it's used to control for the potential effect of electromagnetic field that are known to potentially have an effect on fish. So we decided to include it as well in the experiment. We have two temperature treatments, so 16 degrees and 20 degrees that are the two limits of the temperature that the fish can encounter in the natural environment, upper limit and a lower limit. And we just put that we use heater to warm these three tanks. The noise treatment was changed every week at every trial, but the temperature one was kept at this position. So that's the design. What we did is actually record sessions of 20 minutes each per tank. We recorded simultaneously the acoustic production and the visual behavior in these sessions. We just put a hydrophone in the artificial nest and camera on top to record simultaneously these two features. All right. Now you may ask yourself, what did we get from this video and sound? What are we looking at? Actually, we invested many variables in many of kind. In the behavior ones, we look at chase, dart, approaches, and the number of X. These are occurrences, so it's numbers. We look at durations of different behaviors. The main important ones are of course courtship. I'll show you a bit later what does it look like. Let's display as well when male and female are both in the nest. Yeah, if you want, I can come back to that later after you finish, but it's not so important. And the nest cover, it is a category, so we put either a low category, so it means that the nest is not really built, and the one where the fish build it a lot is big constructions. In the acoustic features, we look at the occurrence of sounds, of course, how many sounds they produce and what kind, the number of pulses, if the sound is pulsed, I'll come back to it as well, and the duration, frequency. In the frequency domain, we look at repetition rate, peak frequency. Peak frequency, they mentioned it earlier as well, is the sound with the most energy and is very commonly used in sound analysis. I can explain it a bit later, after all, if you want and if you are interested in that. So that's here what we are actually looking at. The sound, there is the drum, there is a pulsed sound, and the tom that is non-pulsed. These two sounds, they stay in very low frequency usually. The peak frequency is around 100, 150 hertz, so it's quite low. I would like to show you what it actually sounds like. Hopefully it's working. It's not really working. I don't know if you hear it. Here is the closest one. I don't know if you hear this one. Let's try it again. Okay, so that's what it sounds like. It's not really sexy, but it is what it is. And the courtship behavior, here you can see the nest. It's not the same one as we used, but it's this kind of structure. And here, the male is the bigger one, with the head a bit darker. And right now he's kind of jumping, doing this pattern around the female. When the female gets closer, usually go back in the nest. And it will do this thump-like sound to try to attract. If eventually the female go in, we will move on to the drum sound, and this is what's going to decide if the female lay their eggs or not. Okay. All right, statistical analysis. Yeah, of course. Quick word on that. I use R. I try to use. I use generalized linear model to look for the effect of stressors on binomial variables that are either courtship or no courtship, spawning or no spawning, and sound production, either sound production or not. I use the align rank transform. It's an ANOVA method for non-parametrical variables, non-parametrical data. I use it for the stressor effect on courtship behaviors, the one that I showed before, and sound counts. You can see as well that I have two types of number of data. 53 is the maximum that we use, is when everything is alright. Basically, in this case, I tried to test for the effect with the full data set, but we didn't find anything, so we reused it on the sub-data set with only fish that courted, which kind of makes sense biologically. I use the correlation of Pearson to assess link between variables, between behavior, sound production, and spawning. And I use finally the chi-square test to look at the nest cover, if it has if it's dependent on all these binary variables that I mentioned a bit earlier. And I use a significance level of 0.05 classic. All right, so with all that, it brings me to the results. First of all, I want to talk about the stressor effects. As you can see, nice top spots here. Overall, it was quite mitigated, not so much effect, not so clear actually, but we have some nice and interesting things. First, yeah, blue is the control, light blue is the coil, and red is the noise effect through the speaker. And on the right is 20 degrees, on the left is 16. You can see that the behavior outside nest, so it means that the fish is not going in the nest at all, it's the time data, is significant for control, being higher of the other. And the prediction of thump-like sound is as well higher, but this time for coil treatment, so electromagnetic field. Not significant, but almost is courtship outside is the one that we are most interested in, and is a bit higher for cold treatment. I'll come back to it a bit later. Here, being interesting, not statistically significant, but the time inside the nest, the total time inside the nest by the male, you can see that there is this nice trend of higher values for 20 degrees and higher values for noise treatment. Unfortunately, not statistically significant, but it's still interesting to mention. Yeah, as well, we found out that with the binomial GLM, no significant effect was found on the occurrence of all the behavior, of the binomial behavior of per chip spawning and sound prediction. Here is the nicest result of, it's the nicest result of my project, in my opinion. We find correlation between many variables, for example, between spawning and the time in the nest, the time in which both male and female are in the nest, and the number of drums. We find correlation as well with the number of eggs, the total time in the nest, and male and female in the nest of drums. And finally, some correlation with courtship, this display, and number of toms. I can explain those later if you are curious. Here is what I think is the nicest, is the nest cover alongside probability to display important behavior like courtship, drums, thumbs, and spawning. They're all significant, so it means that basically the higher nest cover will induce more courtship, more drums, more thumbs, and more spawning. I think that's maybe the best one. The acoustic features that I only mentioned was not so easy to analyze because the sample was very low and only 27 fish made sound. Among them, only 15 could be analyzed properly. But we still managed to show that they made longer drums and faster pulses at 20 degrees. Discussion and conclusion now. I want to tell you that we found only a limited effect of the stressors. First of all, no interaction. Unfortunately, that's what we wanted to show, but we couldn't manage to show it. We think that the differences we found in the coil treatment and in the noise in the control treatment were more due to the low number of replicates. Likely that the difference between individuals is what made this difference. Although we think that the coil treatment might be a possible effect of the magnetic field, it's only theoretical. As well, coordination between visual and acoustic courtship support that these fish use multimodal signaling, so they use multiple channels to show their motivation, actually, and that is quite interesting. Nest residence and drop production are hardly correlated to mating, as well, pretty important. And finally, nest building is considered obviously a functional asset, but right now we can manage to show that it was as well a sexual signal, that it could induce sexual behaviors. And finally, acoustic trends, I just mentioned before, that shorter peak pulse period and longer drums at 20 degrees. I just want to conclude by highlighting the key point of this presentation, no interactive effect of noise and temperatures, some very interesting behavioral correlation supporting the multimodal signaling, As well, the temperature could potentially have an effect, maybe with more replicates. And finally, of course, I'm a bit sad, but we need for higher replication and longer exposure, maybe to be able to prove what we wanted to prove in this study. All right, that's it. Thank you. All right.
Okay, so.
All right.
Thanks for an interesting talk. I was wondering a little bit about these trophies. They're usually quite high up.
These ones are relatively flat on the sand.
Yeah, but the depth of it's just a couple of meters.
Couple of meters. I mean, the one we found are this water max, but they can go up to 4, 5, 10 meters. It's usually range 1 to 10.
Yeah. And now I understand completely the temperature, the two temperatures that you want to test. I do understand, but the sound, what kind of, you said something about boat sound.
Yeah.
What kind of boat sound?
I didn't mention this because I had to choose for time, but actually we had 4 playback sounds of boat that we recorded in the Tagus estuary. So it's the river in Lisbon, that's where I did the study. We recorded many samples of boat, not me, but I had the samples. And with this, we made files of boat passing, either ferry boats or small boats, both of them. And we put them in a 1-hour file with a total of 14 passing. Yeah, that was it.
But basically my question, is that kind of something that they will have in nature?
Yes, because they're living up to 10 meters. Like this estuary is maybe, ranging between 10 and 5 meters. I don't know exactly the depth, but I guess, of course, they're facing this. In the Tagus estuary, actually, they've been seen there. So I think they need. Where we sampled them for practical reason was shallow waters, because obviously it's quite tough to go about 10 meters with diving and everything. But they face this kind of boat sound in the way.
I was wondering, in terms of something design, you mentioned it, but...
sorry, I didn't hear it, sorry.
Yeah, you didn't start the question. How did you add some acclimatization time for both temperature and sound before starting the experiment?
Okay.
Did they go direct from stimulation to the temperature? Or the translation was done?
Okay, thank you. Good question. We actually did, not for the sound though, but for the temperature, we did the acclimatation for 24 hours. Every time we move the fish from tank to tank, we give 24 hours of rest to reduce stress. Although once they are in the experimental tanks, they have 24 hours, of course, to acclimate, but then we start the noise treatment immediately, and it goes on for 3 days. We decided 1 hour playing. After 3 days, we cut it and we start recording.
I guess, I don't know, more generally, a second question, but that's just because me, I've been thinking a lot about cycles recently, so I've been focused on that. I was wondering, is there possibly a day-night behaviors into the day versus night behavior into the courtship, and possibly into the stressors as well? Thinking mostly in terms of sounds, I would expect more activity at day, and not at night. So the question, does you expect the sound and the courtship to overlap in terms of time or not? And also,
okay, as well, good question. But unfortunately, we didn't choose to assess anything during the night for practical reason, because we were all day in the lab, and if we have to stay at night to check as well, it's a bit tough. But maybe it could have an effect. Maybe I think this kind of species is more like a day species, as you said, and they would more core during the day. We had 2 big tanks where we would stock them in the first place, and then small tanks on the shelf where we would put them separated. But in the big tank, they were mixed, And we could already see a lot of courtship during the day when we put them all together, and they're actually a species that is courting a lot. It's like not, yeah, we court once every while. If they're courting a lot, and yeah, in my opinion, it's more during the day, although they could do it during the night as well. I didn't really understand into night shift, to be honest.
Okay.
Thank you.
All right, thank you for the presentation. Good to see, I think, combined stressor approach because the reality populations and individuals are exposed to multiple stressors at once. You don't always see these combined approaches. Just a question was about a very basic question about your development plan. You said in the results, when everything was right, we got 53 sample size of 53, but maybe I missed the number of replication or the number of fish to how this was organized.
I went a bit quick on that, but I can actually explain right now. So we had these 6 tanks, and we would do a recording session for the 6 tank in 1 day, and we did a total of 11 replicates of session of a week of trial for the behavior and 9 for the sounds.
Okay.
And then, which means that actually we had a total of 53 individual fish being tested, removing all the potential discarded data that we could have.
There were multiple fish to a tank. So there were multiple fish to, of course, you had 2, right? How many fish to a tank?
No, 1 male, 1 female, and we tested 53 of them. Through.
So it was repeated measures of the same
No, it was every time we fish. I didn't mention that, but we changed fish every time.
Okay, yeah, we were missing this.
Yeah, sorry, but I wouldn't be
really understand.
Sure.
To make
No, it was new fish.
I know you need to make choices. You have 15 minutes. So it's understandable that you can, give just hints of the fact that you're changing the fish.
Oh, you're right to ask, but I didn't think of that. Of course, we had 50, a lot of fish. It was a lot of fish. And we changed it every time for new fish. Although for the female, if they didn't spawn, after 1 month, we reuse them because females are quite tricky to identify first. So as soon as we see a round belly, we're like, okay, this is a female, she might spawn, we keep it. Maybe 1 month we reuse it. But most of the fish were like on.
So yeah, it's very heavy in terms of the lab.
Yeah, no, it was something.
Yeah, of course. And have you 53?
It's 53 male, 53.
Not bad for some people said.
53 female, 53 males.
I don't do experiment, that's why. That's one of the reasons I don't do experiments.
It was something, honestly.
Especially the night. That's all for me.
Okay, so yeah. And questions from the audience? Okay, then you're off the hook. Thanks very much.
All right, thank you guys.
Okay, so that concludes the third session, and now it's a coffee break. So just to wrap up, and all 3 speakers again in this session. Well done.
Okay. Are we ready to start? Okay, so welcome everyone to this final session of the day. As all the other sessions, we will have 3 speakers presenting. They have 15 minutes to present, and then there will be 2 minutes of questions. The jury goes first, and then we'll ask if the public has questions. So I'll start presenting the jury. We have Professor Pierpaolo Bardo from the University of Market, and we have Professor Gautier Chaz from the University of Western Britain, and we have Dr. Esther Blanco from the University of Basque Country. The first presentation is from the team area, and we will go through some metaphoric coding in the other part of the North Sea.
So, good afternoon everyone. My name is Delphine Tange and I got to do my master thesis at the Flanders Research Institute for Agriculture, Fisheries and Food. And it's about optimizing environmental DNA metabarcoding protocols to characterize the fish communities in the Belgian part of the North Sea. So, monitoring fish biodiversity is critical to conservation efforts and achieving good environmental status under the Marine Strategy Framework Directive. And in particular, long-term monitoring is essential for detecting ecological changes over time and assessing anthropogenic impacts such as those of offshore wind farms. And in Belgium, the federal government has delineated an area for renewable energy, being the Princess Elizabeth Island area, which is going to be the world's first artificial energy island. But because the construction of offshore wind farms might alter fish community compositions by introducing hard substrates or due to refugium effects since fisheries are excluded there, it's very important that a before-impact baseline assessment is going to be performed in this future site of offshore wind farm development. And that's why this study is designed to assess the fish communities in the Belgian part of the North Sea, with particular emphasis on this Princess Elizabeth Island area, using 12S environmental DNA metabarcoding. So, eDNA refers to genetic material obtained from environmental samples, and in this case from seawater samples. So in this picture, you can see a carousel with Niskin bottles that were used to collect these samples. And sampling was carried out in the Belgian part of the North Sea, delineated with the black line, in September 2024 aboard the research vessel Simon Steven. So we collected samples at 9 coastal stations, as you can see in green, 4 transition stations in blue, and 7 offshore stations with the ones in red being located in this Princess Elizabeth Island zone, as you can see in light gray. So the dark gray refers to an already operational offshore wind farm. And additionally, next to seawater samples, we also collected beam troll samples at the coastal stations and at the base stations. So more specifically, this leads to the following research questions, with the first one being, which fish species are present in this base zone, so the baseline assessment? Do eDNA patterns differ between coastal, transition and offshore zones? And is there a difference in fish species identified by eDNA metabarcoding and by morphological determination of the beam troll samples? So, how did I do this? As I mentioned, I collected the water samples with Niskin bottles. Then a 2-liter subsample was taken from the Niskin bottles. Then each sample was filtered over a Stereovex filter to capture the DNA molecules, from which it was then extracted and used for 12S eDNA metabarcoding, which allows for the simultaneous detection of multiple species. Then, after high-throughput sequencing, I used the Data2 bioinformatics pipeline in order to produce the amplicon sequence variants, from which I received taxonomic assignments via both Data2 and BLASTN against both a custom reference database, which consisted of 12S sequences of North Sea fish species, and also against the GeneBank database. So the results for this. So to see which fish species are present in the pay zone, I created a Venn diagram, and as you can see, I was able to detect 34 fish species, 2 genus and 4 family level fish identifications with eDNA metabarcoding, and 24 fish species and 2 genus level identifications with the morphological determination of the beam troll samples. Now, and 14 species shared between both methods. Now, some species were exclusively detected with morphology, and that's because some of them had a low taxonomic resolution in a 12S sequence, which is why they could only be identified until family level with eDNA. Then, some other species exclusively detected with morphology actually just had low specimen counts, which is why they were probably missed with eDNA. And then also some specimens were only identified to genus level with morphology, and that's because they had similar morphological characteristics. Then some species exclusively detected with eDNA were actually pelagic species, and so why are they missed with morphology? It's because the beam trawl is selective for demersal species, so that's why the pelagic cannot be detected with morphological determination. Now, since most of these fish species occur in all parts of the Belgian part of the North Sea, I can conclude that there's no unique fish community occurring in this pay zone. But there were detection of some rare species, such as the Alopias vulpinus, the common treasure shark, which has only been detected with eDNA metabarcoding over the past 3 years, with an increase in reed abundance. And this increasing trend is consistent with recent sightings, since last year was the first time that a young individual washed ashore along the Belgian coast. So, with this baseline assessment, I can, again, conclude that there's no unique fish community occurring in the bay, but the detection of some rare species may be important for future conservation and management. So that's why it's very important to continue this integrated monitoring with eDNA and morphology, as changes in fish community may still happen as the development of the offshore wind farm proceeds. Now, some spatial patterns detected with eDNA. So I made an NMDS plot of the Bray-Curtis dissimilarities from eDNA data of the 3 sampling zones, with each point representing a sample taken at either the coast in green, the transition in purple, and the offshore stations in orange. And you can actually see a clear clustering with the strongest separation between your coastal and your transition and offshore zones, which was also confirmed by statistical analysis by one-way Permanova that showed that sampling zone had a significant effect on your fish species composition, with a significant difference between all 3 as confirmed by post-hoc pairwise analysis. So, this can conclude that with eDNA, I could successfully detect spatial patterns in fish community composition, clearly distinguishing coastal, transition, and offshore zones in these well-mixed waters of the Belgian part of the North Sea. Now, to see at the differences between eDNA and morphology, I made NMDS plots of the Bray-Curtis dissimilarities from eDNA and morphological data, so in circles and triangles, in both the coast and bay stations, so in red and blue. And I did this for both looking at all fish species, but also when focusing on demersal species only, because as I said, the beam trawl is selective for demersal species. But in both cases, you can see a clear clustering of both your sampling zones, as I said before, but also of your sampling methods, which was again confirmed by statistical analysis that sampling method also had a significant effect on your fish species composition. Now, when focusing on the alpha diversity, I calculated the species richness in your coast and bay stations with eDNA in blue and morphology in pink. And when you focus on all the demersal species in the lighter colors, you can see that the alpha diversity patterns are similar between both methods. But when you focus on the demersal species only, you can see that with morphology you can actually detect a significantly higher species richness, which probably reflects again the selectivity of the beam trawl. So this was also confirmed by a two-way ANOVA. So with this we can conclude that a complementary use of your 12S eDNA metabarcoding and beam trawl data will enhance your overall biodiversity assessment, since morphology was still very effective for your demersal species, but eDNA could detect a broader range of taxa, including the rare and pelagic species, as I mentioned earlier. Additionally, I also addressed 2 important methodological challenges in eDNA metabarcoding, with the first one being, how can cross-contamination in Niskin bottles be avoided? So, field cross-contamination is a big challenge in eDNA metabarcoding, as it can lead to false positive results. So, this potential cross-contamination could be detected via field negative controls. However, there are currently no standardized guidelines on how these field negative controls should be collected, and many studies fail to report them in sufficient detail. Now, some studies that did report on the negative controls, and they have Niskin bottles that they just rinsed with clean water. Then they collect that water, but those negative controls showed high read counts, so they were not negative. They were actually being an overestimation of your contamination, since just rinsing was not enough to remove residual DNA. So then, this leads to an overcorrection in downstream decontamination steps, which then eventually leads to a suppression of your true biodiversity in those biological samples. So, to address this issue, I conducted a field experiment in which I tested 2 decontamination treatments, with the first one being, I wanted to see whether spraying the Niskin bottles with bleach prior to sampling reduces residual eDNA. And then, whether a 3-minute seawater submersion just below the surface is already sufficient to reduce this cross-contamination. So more specifically, this was the experimental design that I conducted aboard the Simon Steven in April 2025. So we have 6 Niskin bottles. 4 of these Niskin bottles were being contaminated with foreign eDNA of 2 fish species that don't appear in the Belgian part of the North Sea. Then that water was immediately collected as a positive control. Then 2 of these contaminated bottles were being sprayed with bleach, after which all 6 Niskin bottles were being rinsed with clean water and collected, leading to the following sample types: your negative control, bleach rinsed, and the just rinsed samples. After which, all the Niskin bottles were being submerged in the seawater, but just below the surface for 3 minutes, after which they were then being lowered to collect the water samples, leading to additional 3 sample types, the same ones as before, but now submerged. So, then the samples underwent the same procedure as I described earlier, so with the filtration and the extraction, except here they were loaded on the digital PCR to be able to quantify your amount of fish eDNA. And additionally, I also determined the natural 12S eDNA concentration occurring in the water column to determine what are these background levels. And for this, I used 11 eDNA extracts of the samples that I described earlier. So what are these results? Here you can see the average eDNA concentration in copies per microliter per sample type. So with the one in pink being the positive control and showing the highest eDNA concentration. Then when you look at the 3 sample types before submersion, you can see that when you only rinse your Niskin bottles, you have a significantly higher eDNA concentration than when you also treat them with bleach. So this shows that bleach was effective in reducing your residual eDNA. However, after submerging all the 3 sample types, the Niskin bottles, they all showed notably lower eDNA concentrations that were actually comparable to the natural background 12S DNA concentration. So with this, we can actually conclude that this simple seawater submersion is already sufficient to reduce this cross-contamination. So, but however, this still needs to be validated by high-throughput sequencing to make sure that the foreign eDNA of those foreign species that I added are effectively removed. But this would eliminate the need for these unreliable rinse-based field-negative controls that were not effective, and also would not need the use of bleach, which is not very effective on a research vessel. And finally, the last research question: can co-amplification of non-target DNA be reduced by double-sided bead size selection? So several studies have indicated that the primers used in this study that target 12S gene also unintentionally co-amplify the bacterial 16S gene. And as you can see on this electropherogram of a 12S PCR product, you can see the 12S peak, but you also see the 16S peak. We don't want the 16S peak, we only want to have the 12S peak. So I wanted to try to reduce the inclusion of this larger 16S peak to improve my overall data quality by minimizing wasted sequencing effort going to the 16S gene. And I tried this with a double-sided bead size selection, which is a purification strategy using magnetic beads to remove smaller and larger particles, larger DNA fragments, by using 2 bead sample ratios. So for this, I needed to test a whole range of bead sample ratios to see what are the optimal ones. And I could see that a first purification of 0.8 was effective to bind and then also remove this larger 16S fragment, while a subsequent purification with a bead sample ratio of 1.2 was then effective to effectively recover the 12S fragment. So this optimized purification was then compared with a standard purification with a bead sample ratio of 1, that is standard use, and this was tested on 3 eDNA samples. So here you can see the 3 eDNA samples with the average proportion of reads that were assigned to 12S sequences. So with this optimized purification, we expected that a more effective removal of this larger 16S fragment would lead to a higher proportion of reads being assigned to your 12S sequence. But statistical analysis based on a quasi-binomial DLM, general linear model, showed that there's no significant difference between your 2 purification strategies. So yeah, you can see they're almost like they gained the same proportion. So it remains unclear whether this optimized purification could reduce the 16S reads or whether the bacterial loads in these samples was already low. So with my optimized purification, I could reduce the concentration of the 16S DNA fragment, as you can see in my bioanalyzer profiles that I showed earlier, but it yielded no clear sequencing benefits, as you can see here. So that's why more research with more sample types is going to be needed, and preferably from areas with a high bacterial load, so that you can see two peaks. So yes, by both optimizing field and laboratory protocols and contributing to a growing environmental DNA dataset, this study supported the development of standardized and effective long-term monitoring strategies to safeguard the good environmental status of the Belgian part of the North Sea. So thank you for your attention and thank you for listening. Thank you also for my supervisors who are probably listening right now for your amazing guidance through my whole process. Thank you.
Well, thank you very much for the presentation. It was quite clear and I like it. So
Thank you.
well, just two curiosities. For example, do you know the friend that we use,
Yes.
12S?
Yes.
Which protein or part of the DNA corresponds to?
It's the 12S mitochondrial ribosomal DNA gene. So, yeah.
Why did you select that?
Because this one, so we use the MyFish primers for this, so they target this. It's a universal primer, and it's been shown in studies that this is like the most used primer and also like target gene for assessing fish biodiversity, and even in freshwater, also in seawater. But it's been proved to be very effective and really used a lot. But then again, I also showed that it has this 16S amplification, which was now addressed for the first time. So yes.
This project, the master thesis is inside of another bigger project in which this optimization will apply?
So the two optimizations that I did are part of two bigger projects. I mean, I work together with a PhD student, but I mean, they just need actually my results of the high throughput sequencing to be able that other people of my institute are able to publish, because it's a big problem, this field negative control, which is why I had to address it. But the co-amplification test is not really necessarily linked to another big project, but I know that the cross-contamination test is, I mean, they need the results, but it's not part of anything like another really project, you know. Yeah. Yeah.
Well, that's all from my part, and thank you very much and congratulations.
Thank you.
Okay, I'm just wondering about the introduce to clean the risky compost and prepare for EAPA conversion. Because you know, when you deploy a CTD system,
It's what?
A CTD system with the risky bottle, you need time for the integration of the part, and the CTD system is at sit for minutes before the activation when it's circulating in the circuit of the sensor. Installed. So, do you think that this time is, I don't know if you never be on board when doing a CTD cast, I don't know, but it takes time, it takes minutes. So, do you think that this time can be enough to ensure the cleaning of the
3 minutes?
Yeah.
Well, yeah, because this is also like standardly done to calibrate the CTD, so the conductivity, temperature, depth. So it's already included in time for sampling, because as you said, like it takes a while, like to take samples everywhere. So this is actually already a bit included, but this rinsing with the bottles open was not done before. So it's just now something that we include. Do you know what I mean? So it's like, it's the calibration of the CTD that's also done with a 3 minutes submersion already. Yeah? Okay.
Thank you for your presentation. I think that is really, really important for two reasons. I really like the comparison between the morphological and the DNA approach, which is producing, and also the the optimization of protocols, which is something that is often overlooked, and as you say, it's getting more and more mandatory for publishing, and it requires that kind of attempt to be done. I found really interesting. So, first I would like to ask you about the comparison between the two methods. So you showed an NMDS with the results from morphology and from eDNA.
Yeah.
Well, unsurprisingly, there's a difference to show us that the species identified were different, so there's no surprise here. But did you try to run an MDS or any sort of equivalent to show if morphology highlighted the same pattern, the same spatial pattern that eDNA would show the...
Also, to see there's also spatial patterns
Yeah.
with morphology.
Yeah.
I didn't make a separate NMDS plot because I only have beam trail samples taken at coastal and the base station, so without transition. But so actually, you just mean
I can see from here that...
yeah, you just mean like by excluding this if these still show the difference, right?
Yeah. And
Yeah, it would clearly still have the clustering.
And did you have a look at which species were driving this spatial pattern?
Yeah, so also a bit here in the Venn diagram, as you can see. Oh.
The spatial pattern. Yeah.
I want to, you mean which ones appear in the coast and is transition offshore?
And if the two approaches give you the same information.
No, I didn't look into that separately, but previous studies have done it with like also having them in the coast, transition and offshore, and then they make this simper test to see which ones are present, like determine this is how we can see in the coast, these ones are more here, and the transition days, these are here, and the offshore here. So, but I didn't do a simper analysis because I didn't have to do it, so now I can give you the answer to which species are more occurring, determine this spatial distribution. So now I didn't do that now, but it has been done before.
And if I understand well, for your experiments for decontamination, so you did artificially contaminate the skin bottle with DNA from two alien species. How did you do that?
So the two foreign
rotting fish.
No, but it's a very good question. So here you can see the two species. This is actually Pseudochromis pitmani and Sparozorata. So these were actually two fish species that we had present in the aquaculture lab of my institute where I was. So with that I did analysis they're sure that they're not occurring here. Well, this one is a freshwater species occurring in the Red Sea, and this one is only occurring in very low abundances in stations that were far away from mine. So how did I do it? So we had tanks with Pseudochromis pitmani and another tank with Sparozorata. So in the morning before I went on sea, I really took water from their tanks and I put it in, 2 liters in total, and then I just poured the water bottle in the skin bottle and immediately collected it. So, but it's fresh eDNA, as I did it in the morning but I also, before I took these two species, I did lots of experiments in the lab to make sure that my primers work on these, both separately, both together, I did a lot of things to make sure that they work, and then also the positive control here shows that it works, otherwise I wouldn't have any application.
Okay, thank you very much.
No worries.
Okay, now if the public has some questions, this is your chance to ask. No? Okay. Are there urgent questions from the jury or? Okay, the chat urgent. Yeah, there's no questions. So, congratulations. Thank you again. And now from the North Sea, we move to Antarctica.
Finally.
With the presentation by Sarah Meyer. Yeah, okay, perfect. You can start when
I can start. Amazing. So thank you for listening to my presentation about what I did the last half year. And it has the title of Disentangling the Structure of an Antarctic Plankton Food Web in bloom and non-bloom conditions. So what is it about? First of all, plankton, especially phytoplankton, as you can see in the background here, is the basis of the food web, as it produces all the energy for the higher trophic levels as fish and such. And earlier studies in the study area showed that there's actually a lot of variation in phytoplankton. This is chlorophyll A as a proxy for biomass over the last decades. But at the same time, food web studies done in the same area sum up the phytoplankton in very broad boxes. So this is a food web for all the specimens, but it has only 3 phytoplankton nodes. 2 of them are benthic and 1 pelagic node. So this led me to the aims for my work of to disentangle the plankton boxes in the food web for a better understanding of the energy flows and to better predict the impact of environmental changes on the system. My methodology in a nutshell. I got empirical data, so observations from past work for a non-bloom and a bloom year for phytoplankton and zooplankton. And then I used those observations, so the species list that I got, to do a literature review for the interaction, so who eats who. I combined this data to create an unweighted food web with all the possibilities of energy flow within the plankton, and did like standard food web analysis. And as a next step, we added weights to the interactions and filtered again for the non-bloom and the bloom year to compare the 2 states. So the study area is Potter Cove, which is at King George Island at the West Antarctic Peninsula. More concrete, all my samples are from station E2, and they're all from the summer season as a seasonal average. As said, I have a bloom year and a non-bloom year. Those were defined in the work of Anthony et al. Published last year. And this is important to keep in mind, since bloom year does not have a threshold to non-bloom year. It is a little bit relative to the other year, what amount of biomass is needed for it to be a bloom year. And I have phytoplankton and zooplankton data from different works that have been done before. So they were not collected at the exact same day, but they were collected the same amount. So I have roughly 5 samples for each season for phyto and zooplankton. How did I get the interactions? I followed a systematic literature review with the approach of Odea et al. That means I defined a search term and I put it in 3 scientific search engines named PubMed, Scopus, and Dimensions. And then I filtered for duplicates in the DOE and title combination and removed the duplicates. That left me with 1,738 papers to screen through the titles, and if they sounded interesting, the abstracts as well. And in the end, we used 115 papers that contained information about the stomach content, observations, or the fecal pellets that gave us the information about who eats who. The next step was to assemble the food web. This happened first by data cleaning, so all the data we got from the interaction, from the search for the interactions was like cleaned, the standard typo removal, and also for example, detritus was like a lot of different names in the papers, and we all said that's detritus. And the next step was actually quite crucial. It was the aggregation of the species into trophic species. Because since it is a food web analysis, we use trophic species. So multiple biological or taxonomic species can actually be one species if they have the same trophic niche, meaning they have the same set of predators and prey. And this is important to avoid redundancy in the analysis. And those groups where there is no methodology for those groups in the phytoplankton and also not in the zooplankton. So we defined them with the help of experts, namely Gaston Arnados for the phytoplankton and Maximiliano Garcia for the zooplankton, which are experts in the area in phytoplankton and zooplankton respectively. And our proposed groups are defined based on taxonomy, shape, physiology, and size. And we came up with this classification because on the basis of the data that we had available and what we wanted to do with it. And this is the food web. It was pretty cool to see it, and it consists of 37 trophic species and 213 interactions between them, which leads to a linkage density, so a mean amount of interactions per species of 5.76, and a connectance value of 0.16. Connectance is the amount of interactions relative to the amount of possible interactions, just considering the amount of trophic species. If we look at how the species are distributed, so there are only a few top species, so only a few species that have no predators, but more than half of all the species are basal species, meaning we consider them autotroph. And this leads to a relatively low trophic level of 1.65. The definition of a trophic level is the distance of the species to the basal species. And the omnivory level is 0.41, meaning that 41% of the species eat or have prey from more than one other trophic level. Generality and vulnerability also refer to prey and predator. So a generality of 12.52 means that each species on average has 12.52 possible prey species, and each species with a vulnerability of 6.9, each species has 6 possible predators in this system. Modularity is another possible another analysis you can do with the food web, since it is assumed that a network is always organizing itself into sub-networks that have a higher connectance and more interactions within the subgroups than between the subgroups. And the modularity for the plankton food web is 0.17. And if you have those subgroups, there are 4 topological roles defined based on if there's generally a high level of interactions, if those interactions are mainly within the module or between the modules. What is interesting to see is that there are no module hubs and no network connectors, so all species are considered to have a low amount of interactions in general, and there's 23 module specialists and 14 module connectors, so the majority of the species have more interactions within their module than combining modules. The degree distribution is an analysis of the interactions, so this is the frequency of the interactions, and then we fitted models to this distribution, exponential and power law and non-uniform model, and the exponential has the best fit, meaning that there's a lot of species with only a few interactions and only a few species that have a lot of interactions. Now to the second part of my analysis. So we had data from a bloom and non-bloom conditions, and we wanted to get the weight of the interactions, and for that we would need biomass data. We only had biomass for a few phytoplankton species, so we followed the methodology of Kara et al. For the estimation of the interaction strengths. It is important to note that this methodology does not actually calculate the energy flow. So it's not a measurement of carbon, but it's a relative measure of, okay, there's more energy flowing in this interaction than in the other one, in the map. And so interaction strength is defined as the effect the prey has on the predator. For this methodology, you need the density and the body mass. And the body mass for my data set was given in carbon content per individual, and we converted it with conversion factors from the literature into wet weight per individual. And for species that had no body mass values given, we used the body mass values from the Weddellsey data set. And then we filtered the whole data by density. So for the bloom year, everything that had density higher than 0 was included, and the same for the non-bloom year. And those are the two maps. And you can see on the first glance that they are a little bit different. Specifically, the bloom year is smaller with only 22 trophic species and 102 predator-prey interactions, while the non-bloom year has 26 trophic species and 126 predator-prey interactions. And if you look at the weight of the interactions, it is quite it's quite easy to glance that all the heavy interactions here in yellow are connected to only a few points. For both years, they are connected to detritus, and in the bloom year, they're also connected to the euphausids, while in the non-bloom year, there were no euphausids observed. If we compare the properties for the two scenarios, we can see that the bloom year has a higher property for basically all of the values. They're the same but calculated with the weight of the interaction. The only difference to the unweighted measures is the omnivory level, because this refers to if the species eats from different levels, but mainly, like all but one prey are from the same level, or if actually all of the preys come from a different trophic level, then the omnivory level would be very high. If we look at the weights compared to the trophic level, like the total weight, so all the interactions within one node, and we compare it between the years, there's a few things that we can note. The first one is that, so this is organized by trophic level, with the highest trophic level up and the trophic level 1 down here. The first thing that we see is that starting with predatory species, there seems to be a connection between the trophic level and the weight, because it seems to increase with trophic level. The second thing that we see is that the higher trophic levels, there are some species that are only present in the bloom year, while in the trophic level 1, so the basal species, there are some species only present in the non-bloom year. And the third thing to observe is for the species that are present in both years, most of them are actually quite similar, but for example, the blooming species, the large thalassioceroids, are significantly different, or probably significantly different for their weight based on the box plots. To my conclusions. So this was the first time the plankton was disentangled to that degree, and it's pretty cool to see that it's actually possible to make a plankton food web in Antarctica. Then the unweighted food web is the map of possibilities, but it does not actually resemble the actual state in nature, since plankton is that variable and system with dynamics that are really fast. And we could see that because the scenarios were different and not the same. The next thing is that in the bloom scenario, we have higher trophic level, which leads us to the conclusion that there's probably more energy available for higher trophic levels that were not considered in this study. And the last thing that was pretty interesting to see is that in the bloom year, we had krill, while in the non-bloom year, there were salps, but not the other way around. And combined with the lack of higher trophic levels in the non-bloom year, it can be considered that salps are a dead end for energy as they are considered in literature. So I thank you for listening, and I'm happy to discuss my work with you.
Okay, well, thank you very much for the presentation and congratulations. Well, just the zero data you manage, I suppose, in order to perform that analysis. So, well, what was like the hardest part of that?
For me personally, the hardest part was to actually get all the data assembled, because the majority of the time I spent in the last month was assembling the databases. We got the data for the zooplankton and for the phytoplankton, and it was in different units, and especially for the weighted methodology, I needed it like the biomass or the density and the body mass, and there was a lot of literature research involved, and getting it all together into a state that actually was usable for building a food web, I think was the hardest part for me, yeah.
And what would be the further research that you do?
The further research would be to, for example, use, in this case, we compared a bloom year and a non-bloom year, but if we look at Potter Cove, we can see that there is, like, the bloom years are mainly very cold or very hot years, and then in those two bloom scenarios, the phytoplankton composition is different. So further steps would include like other variables like temperature, and actually look if there's a difference between the different temperatures, or also compare within 1 year, like before the bloom, and after the bloom, to get a higher temporal resolution, since plankton is just a very dynamic system. Another option would be to try to get actual biomass measurements for all the data to estimate the actual energy or carbon flow between the species. So there's a lot of options to go forward from this.
Thank you very much. It was a very interesting presentation, especially talking of the food web ecologist with a major interest on non-organic organisms.
Yeah.
It's been quite a while that everyone agrees to say that plankton is a black box, and just like plankton, there's a whole world taking in.
Exactly, yeah.
And that's super interesting to see that some works are trying to decentralize what's going on in this course. I really appreciate that. So my first question is about your literature review, finding information. Did you manage to find enough information in this literature?
It was hard.
Especially on the right taxonomic resolution and for the. So I guess you have to extend the area to.
Yes, the area was extended to whole Antarctica, and that was also a reason for aggregating the species, because obviously the taxonomic resolution of stomach content observations is not on the species level of the phytoplankton. And also for some zooplankton species, there was like a lot, okay, this eats mainly phytoplankton smaller than 20 micrometers, or for the tintinids, it's dependent on the lorica size, but then it eats basically everything or stuff. So there are some assumptions involved, but for the majority, we found interactions. Yeah.
Okay, super interesting. How do you explain the fact that you found the lowest trophic diversity in non-bloom conditions?
Here.
Yeah.
The trophic level.
Well, the diversity and the lower mean trophic level, the lower connectance, lower the
Yeah.
it's all lower in non-bloom conditions. Is make sense to you?
Yes, because in the non-bloom conditions, you can actually see it here. We have a lot of species that are basal species, and basal species have interactions not with each other, but only with predator species. So if the percentage of basal species is just really high, trophic level will go down, and the other fluid measures probably as well, because to keep connectants at the same level, there would need to be a lot more interactions within the higher trophic levels compared to the other scenario.
Okay. And there's also a result that I found interesting that all the metrics for bloom condition suggest that we have a community that is probably regulated by top-down processes, especially microtrophic level. Do you agree with that or not?
So that the processes are top-down controlled.
Which is not really tricky for bloom.
Yeah, that's hard to say actually. I'm not. There are studies that assume that a bloom condition is possible when there's a decoupling with the microzooplankton. Since microzooplankton grazing keeps the phytoplankton abundance or density at check. So there is actually a top-down control assumed for bloom conditions in Potter Cove at some level. But I wouldn't say that it's all top-down controlled, since phytoplankton blooms, there's other works that show that they are mainly related with temperature, and in Potter Cove specifically with higher temperature, there's like more glacial melting, more turbidity and that stuff, and that keeps phytoplankton or influences a lot the phytoplankton. Yeah.
It kind of looks to me like a trophic cascade. But you're observing the fact that you shift. That's a big difference. Shifting from 1.93 to 1.5 is huge. And you know, when you add a trophic level to a food web, usually you just completely shift the biomass at each trophic level, and that's something that could result in what you're observing. Normally, when you have the species on top of the food web, not when you modify what's going at the base. So that was super interesting. I hope I had an explanation also that just wanted to be able to fast.
Okay.
But yeah, thank you for the result. Super interesting.
Thank you, very nice presentation. Question from my expert in the field. But you got your results not based on two conditions, room and room. And you analyzed a huge number of applications. Is that right? Is it correct?
For the interactions, you refer to literature review. But this literature review was really focused on the interactions. I didn't read anything about trophic information other than. I spent 4 weeks only on screening the papers. The timing I used. So I got the observations. I'm not sure how long they took to identify the species and sample. I don't know that. I got like Excel tables with, okay, we found this species with that density, and for some of them the biomass or the carbon content individual. And for the literature review, it was, I think 1 week of defining the search term and like getting through the databases to extract all the papers, and then 4 weeks of only reading the titles and if they were interesting, the abstracts, and that. Is that what you wanted to know? Yeah.
Over each time interval, estimated and observed for when?
It was the summer season. So from January to March, so Antarctic summer, for 2014 and 2017. For 2 years, yes.
And so you can get information about availability of your composition?
No, it's not about temporal variability. It's just a snapshot and another snapshot, and then a first comparison. Since in the beginning, the main focus was really to assemble the plankton food web and see if it works and how it works to get the trophic species, and if like the values we get out of it make sense, and then to compare. Yeah.
Would you like to carry out, or do you think to carry out this analysis in other areas?
It would certainly be possible. At the moment, I would like to finish my master thesis first.
Thank you very much. Is there someone from the public who wants to ask a question? Okay, then thank you very much. We're a bit early, so I will wait 10 minutes for the people online so that when they go.
Okay, perfect. Well, it seems like I need time to get out of this thing, so is it not just oh, maybe it's the control. Yay! No. Do it there. Ta-da. Fabulous. And I'm just gonna close.
It should be open, yeah.
Yes, it's open.
Yeah, I would say it's 10 minutes because we're
Oh, we're that early. Okay, perfect. So
just for the people. Do you know if there's someone who's coming to see you on that answer?
I mean, if they are, they can do it later too.
I guess if they were waiting to see you,
Yeah,
but
that's
if
true.
connect
I can
and you're
check
already
if anyone
done,
said
you know what I
anything.
mean. No, but it's okay. Yeah, there's also those other people in supervisors. Oh, maybe. Okay, good. So from one pole to the other one, now we go to the Arctic with native species. Presentation by Victoria.
Okay. Hello, my name is Victoria Ronneberg, and and today, in our final presentation of the day, I will be presenting my work on biological invasions in the Arctic, which was under the supervision of my supervisor, Melina Pinotidou, at the IUN Amur Lab in Brest, France. So, invasion science and invasion biology is filled with terminology, so we start there. Biological invasions occur when organisms are transported and released into areas beyond their native geographical range through human-mediated processes. And upon their establishment and further spread, these so-called non-native species are able to cause dramatic and irreversible changes to ecosystems across the world. Today, they're considered to be one of the major threats to biodiversity globally. With increasing pressures from warming waters due to climate change, increased human activity due to receding ice, as well as range expanding species from the boreal south, the Arctic is rapidly transforming. And this is increasing both the likelihood of non-native species introductions, as well as the likelihood of their successful establishment in this region. So, although there is collaboration across Arctic, in order for us to be better prepared on this inevitable change, we first need to understand what the current situation looks like. Due to sparse populations, limitations in access, and high costs in research, the Arctic remains largely understudied, and this is particularly true for the marine Arctic. And obviously, the lack of information makes it difficult for authorities to make informed decisions on regulation and policy actions. But we can argue that although data is limited, and because it is hard to retrieve, it is even more important for us to maximize its value by looking at it in different ways so that we can extract as much meaningful insight as possible. One of the major challenges to analyze data and identify trends in the Arctic is simply that the Arctic is very large in comparison to the data we have. So to overcome this, we need to be able to look at our data through some alternative lenses. The Arctic large marine ecosystems were delineated by the Arctic Council's working group, COSF, the Conservation of Arctic Flora and Fauna, in 2013 and then updated in 2015. And for those of you who are not familiar with the large marine ecosystem framework, it's a way of dividing the ocean into regions based on ecological and oceanographic criteria, thereby capturing some of the complexities and dynamics of marine systems and allowing for ecologically more meaningful research assessments. And while large marine ecosystems capture the ecological dimension, it is also important for us to include jurisdictional boundaries. As you may know, under the United Nations Conventions of the Laws of the Sea, the regulation and policy power and rights remain largely within the Arctic coastal states' exclusive economic zones. Incorporating this layer as well to the analysis will give us some clarity on where actions can be taken and by whom. Altogether, this forms the research question that I've been working on, which is, how does the current distribution, status, and documented impacts of marine non-native species across Arctic large marine ecosystems, exclusive economic zones, and international waters inform our understanding of regional vulnerabilities, monitoring gaps, and opportunities for circumpolar management collaboration in the region? I understand, it's 5 o'clock, it's a lot of words. So, to simplify, the hypothesis is that by looking at our data through these 3 lenses I'm going to talk about today, which is distributions across large marine ecosystems, distributions across the Arctic jurisdictional regions, and what we have from documented ecological and economic impacts in the Arctic and nearby regions, we may be able to combine the findings and thereby uncovering some new trends. Data-wise, I produced 2 separate data sets. Dataset 1 focuses on species distribution and was constructed by extracting species distributions from the Global Biodiversity Information Facility database based on a predefined list of global non-native species. This was then overlapped with the spatial polygons in R of the large marine ecosystems and EEZ in order to be able to classify the existence of these species across these spatial parameters. And additionally, I had to include status classification data wherever it was available in order to be able to exclude any native occurrences, any species that were occurred within their native boundaries. What we're left with then is non-native, not exclusively native species distributions across the LMEs, their distribution across the EEVs, as well as their status classification within these spatial parameters. Dataset 2 focuses on impacts and is based on the list of identified species from dataset 1, that is, the species we know are present in the Arctic, and was created through a systematic search of ecological and economic impacts of these species within relevant databases. These are highlighted here. I also noted the region of impact as well as what I call the evidence level, which is if the impact is based on documented observations and quantitative data, or if it's more indirect connections and speculative impacts. Because this is an exploratory investigation and we're trying to be detectives of biotrends, several analyses were done, some of which are listed here. Due to limited time, today I will only focus on findings, results, and dimensions of the spatial analysis. This is because this aspect is most contextual and fundamental to understand any sort of trend. So, over to the results. In terms of the distributions across the large marine ecosystems in the Arctic, I identified a total of 409 unique species, and they were present in all Arctic LMEs. In this graph, you can see the total number of species in each LME in yellow. And if you notice, the distribution of the species was not evenly across the area. The highest occurrence was found in the Norwegian LME, Norwegian Sea LME here, as well as the Barents Sea LME. These two also have the highest species density when you normalize for area, in addition to the Faroe Plateau LME, which is this tiny number 1 here. When we look at the data through the lens of the jurisdictional boundaries, we find that trends are similar to those of the LME trends, with hotspots in the Norwegian EEZ, as well as the Svalbard and the Jan Mayen EEZ. Importantly, we also identified, although it's not present in this map, we also identified 29 species to be present in international waters. And in international waters, I mean the high seas of the Central Arctic Ocean, the high seas of the Norwegian Sea LME, as well as the Barents Sea loophole, international waters. So, I won't go too deep into the impacts, but what you need to know is after an assessment of 145 species in these databases, we identified ecological impacts in northern regions, that is, in the Arctic or Arctic approaching regions, for 25 species. This includes the Atlantic rock crab in Iceland, which is found to outcompete native spider crabs for food, ultimately resulting in a general reduction in the body size of native spider crabs. We also have the pink Pacific salmon in northern Norway, which during spawning season floods the river systems and alters the dynamics of the food web by becoming a major seasonal food source, both for animals in the water as well as on land. When these ecological impacts become severe, they often translate into economic impacts. When we found out of the 409 species that we identified to be present in the Arctic, I could find monetary cost estimates for 10 of these. Only 10. That is less than 2.5% of all the species we found. But just these 10 had cost estimates that amounted to $186 million in 2017 exchange rate. So, from this very short glimpse of the impacts data, what we can say is that the species that we have found to be present clearly are capable of having impacts, some of which have already been observed, and also that the impacts that we are seeing are likely underestimated. But what does all of this tell us about regional vulnerabilities, monitoring gaps, and collaboration potential? Well, firstly, no place is safe from invasion. Even in the remote areas of the Arctic, we found species to be present. So that means no area seems to be untouched, no area seems to be incapable of having new species appearing and establishing populations there. Secondly, the hotspot we found seemed to align with human activity. If you can remember, majorly the main species densities were found in the Norwegian, West Russian, and Northern European coastlines, and these are also the areas with the highest population in comparison to the more remote northern coast of the Canadian, Russian, and Alaskan coastline. And this information gives us, we don't know if the variance in species density is caused by simply higher introduction rates because there's more human activity, or that we're just monitoring these areas better and thereby finding more of the species, which is, kind of an important factor to answer. But if we pull the argument further and we argue that human activity is the main driver of why these species are so dense in some areas, it would be beneficial for us, knowing that the Arctic is becoming having more activity. It would be more helpful for us to put more monitoring pressures in the areas with low data where we're expecting human activity to grow in order for us to have a better baseline to compare future risk assessments to. Yes. And although we may know a bit better where these species are located, our impact assessment shows that we know very little about how they're actually impacting their new environment, which is a crucial step in order to make management and regulation targets more targeted and more efficient. In terms of collaboration potential, I want to highlight 2 points. One is the fact that because we found species present in international waters, it highlights that this issue is not just a national issue. Species are crossing borders, and so is the responsibility to manage and mitigate the consequences. Therefore, future work in the Arctic into species assessments should be a collaborative effort. Finally, as this project has tried to show, is that by looking at data more holistically and taking several analyses into play, looking at several lenses, we may be able to uncover trends that no single nation or single LME would be able to spot by themselves. In other words, the whole is greater than the sum of its parts. In conclusion, the paper shows that Arctic invasions are more widespread and complex than we previously thought, that pan-Arctic collaboration is vital to understand risk, highlight knowledge gaps, and furthermore protect the Arctic environment, and also that this thesis is an example of how a connected and informed approach may identify potential trends and also risks in biological invasions in the Arctic. I also highlighted some future research priorities, which I found. One of them is to investigate variations in regional monitoring activities. If we know the monitoring level within each region, it's easier for us to incorporate that data and make more sense of our analysis. Secondly is to improve the status assessment for data deficient taxa. If we don't know where the species are from originally or what they do, it's hard for us to make that information useful. And finally, with increasing human activity in the region, although it provides a greater risk to introductions, you can also look at it as a greater opportunity for better monitoring efforts. So finally, I would suggest the idea of a preliminary investigation into how citizen science are using these new human activities as part of gaining more information about the issue. Thank you very much.
Okay, so congratulations for the work, Victoria, and the presentation very clear. But when I read the title, I expected more socioeconomic impact. But I saw that this master is more biological, but was very, very good.
Yes, yes.
Was, as a curiosity, in the final part, we talk about the economic impact of all of this, and say,
That is a huge amount of money, yes.
Yeah. So I was curious about maybe, how those calculated this number.
Yes, so that the economic monetary estimation system is based off a database created called InvaCost, which is a compilation of all cost estimates produced for biological innovations. And it's based on, it's a standard, it standardizes the cost estimates and also classifies them according to if it's an observed or predicted cost. It also classifies them according to what sector is impacted and also what type of cost. So in my case, all the costs were either related to damages or management. And yes, did that answer you, or was there anything?
No.
Yeah.
Also, in the introduction and business, university, you talk more about the classical species area nowadays, and you can give some examples of what is the main origin of those species, about that information. Can you tell us?
Well, the most studied species are basically in the Barents Sea, and it's especially the red king crab, which was introduced by a Russian to make a crab fishery, and has then spread, and then you have all this legislation connected to that. We also have the pink Pacific salmon, which is the same thing. It was also introduced to start a new fishery. But other than that, studies on invasive species in the Arctic are very limited, and they're still trying to uncover. Like the most recent, the greatest compilation was made this year by the Arctic Council, and they're still working to define the species list. Even though the general report is published, they're still updating the species list. But it ranges from algae to fish in general. Thank you.
So, just a question really about the reason of this invasion. Can you any evidence about if the maritime traffic due to the sea ice is declining in the Arctic, so there is much more maritime traffic?
Yes.
Can be a source of this invasion?
Definitely. They're doing studies on this now, and I think, especially like notably in the Norwegian Sea, they found a 47% increase in ship traffic, and that includes both, transportation, but also fisheries, tourism, and generally in the world, shipping is considered to be a major vector and introduction pathway, both in terms of ballast water, but also in terms of like biofouling, that the species are connecting. So I didn't have time to go into comparative statistical analysis, but part of the information on boat traffic is being updated, and so that would be a nice future investigation.
And is there any scientific evidence of the role of the ocean dynamics in transporting matters into this area?
Into the Arctic? If there's any studies on if ocean currents are bringing species into the Arctic specifically, there probably is, but I haven't gone into that deep, so I don't want to say anything specific. Generally, it's like that species are moving north, or that you have these great shifts. I don't know if you read the big mackerel population also did like a big shift one year. So yeah, in terms of fisheries, which are the most studied species, of course. But the larvae themselves, I'm not quite sure, so more time for that.
Thank you very much.
Thank you, Victoria. That's a really interesting topic. I have to admit that I don't remember during my life asking myself invasive species in the Arctic. The thing is that there's a reason for that. I think I was thinking about the Arctic as a particularity regarding invasive species. It's that I think that 100% of invasive species in the Arctic come from the Arctic. I don't know, I don't have any example from Antarctic species being introduced to the Arctic.
If I have an example of an Antarctic species that's been introduced in
So I think all these basic species come from the Arctic and they've been introduced to other parts of the Arctic.
Yes, that is very common. And we saw that out of the international species, they're all also present in other Arctic species.
Yeah, there's a reason for that, that the Arctic is connected to all the oceans of the world. And so the Arctic is going to go through major changes in the decades to come. It's already been through some major changes because of climate change and range extensions that will result in temperate species invading the Arctic at that. Because it's range extensions, it's not exactly invasive species. So these are only examples where there's introduction and we get further examples. I have to say I was surprised that we think 400 species. And I wish I had the list of those species.
I have the list.
Yeah, I'm sure you do. It didn't come out to be like that. Nice number. So, do you have any information on the vectors of introduction?
No, I didn't include introduction vectors as part of the scope.
Yeah.
I focused more on the species presence and the temporal trends and the status. Like, much of the research was actually based on like how much we don't know. Like, when you classify the species based on like if they're considered to be introduced, range expanding, invasive, likely native, generally it took a long time to calculate around 409 species. So I didn't have time to look into introduction, and I also feel like that's an area that much of the research is kind of aiming for.
Yeah.
So. But I know that they majorly highlight shipping, like ballast water and biofouling, and to some extent aquaculture are considered to be the major. And then you have these other.
Yeah, like you mentioned the Atlantic rock crab at some point.
Yes.
What species is
The Atlantic rock crab.
Yeah.
What the Latin name is. Can I please come back to you? What the scientific name for the Atlantic rock crab is? Can I please come back to you on that? But I don't know the Latin name by heart, and I also don't have my notes.
I'm super glad there's so much names, because if it's an Atlantic crab, then it has to be just an extension range of its range. So it's not probably non-native species.
No.
So that's why I was.
It's a very.
Curious to this list of species. I'm pretty sure if we look deeper into it, some of them are not the species.
That's a good. Can I just
it's gonna go through some rough time.
Yeah. There's also a lot. So the list I have is just species that are not classified as native.
Yeah.
So in many cases that would include range expanding species that haven't been there, like doesn't have evolutionary history there. It includes species that are what's called cryptogenic, which are the species that we also don't know where they're native and they were recently discovered in the area. We don't know if they've been there the whole time or if they come in from somewhere. And that's a major limitation. So just for simplicity, I use the word non-native species to describe this because terminology is always a bit tricky. But you're completely right. Out of the 409 species, not all of them are, like very few are actually classified as introduced. But it's more of a baseline, I would say. Like it's a starting point to do that investigation.
That you have to start somewhere.
Yeah. No, but it's a very good point. Thank you for highlighting it.
I really appreciate the fact that you question that spatial pattern between the number of invasive species.
Definitely.
I would have been really surprised to find the highest number on the region this time. And I would have guessed Norway for sure.
Yeah.
to remove
very
guys. Okay, yeah, I think that's pretty much it for me. So thank you very much.
Thank you.
Does the public have some questions?
Also, go get some drinks.
Well, thank you very much for this insightful presentation. I asked myself, is also the number of species that were activities that cause these interactions, but also I think like if there is a lot of data in one region, it is likely to find more species. If there is like some kind of connection to this, or if there is some data that I don't know, I don't have the answer on how to relate these two kind of that I don't know if you've done something in this direction.
If I'm understanding you correctly, you're talking about how the monitoring gaps, like that you find more species in some areas because they're just more investigated? Is that it? Yes, so that was my priority for future research, is to actually identify, like the first thing we should do is actually get an overview of what areas are monitored and to what extent they are, because or else it makes it seem like Norway is just very invaded by species, and that invasive species just don't like Russia and Canada a lot. You know, like, so that would be a first. I haven't gone into, I think that would require another master thesis to go into what monitoring activities are done in each area, but definitely.
It also might be a question of who is openly sharing their data, because it's like Norway might be sharing the data and Russia will not, and that might end up distributing directly.
Yes.
It's not a lot of it's just like curiosity.
No, definitely. That's definitely a point, that the data access will be different, and we also don't know what the different countries are looking for. And it would also be connected to that, you know, like if there's one type of, if it's usually registered as part of like bycatch and fisheries, then it would be dependent on what type of fisheries you have and where you're looking, and if you're doing benthic surveys or not. And so I think all in all, the conclusion is that we know little. And there's a lot of uncertainties. But yes, good point. Thank you for the question.
Okay, thank you very much, everyone. Thank you.
Allora questo quanto sarà costato?
Che?
5 euro.
Vai.